progress report from bench to bedside. Thank you so much.
Dear Chairman, thank you very much for your kind introduction. It's my pleasure to be here, dear organizers. I thank you for allowing me to speak here. And I would like to go into a little bit more detail, since some of you may remember my presentation of yesterday, to explain the idea of what it means to activate, reduce regeneration inside the human body. Now, as you can imagine, the stem cell technology has brought about a revolution in the field of biomedicine, and it was predicted that it may be a wonderful time, but the ride may go to be wild, because a lot of changes have happened and the first clinical results are now obvious, and many technologies have been developed. Now, it started from embryonic stem cells, and embryonic stem cells, like a human being, starts just from 1 or 2 cells and becomes a living human organism, and different tissues are differentiated in the human being, like skin, neurons, or liver, for example. However, if you think about cell culture systems, cell culture is a complex way of preparing tissues, and the demand of time to create the tissues from a cell culture is several weeks, maybe 6 weeks, or 4 weeks, or 2 weeks. And the logistics is relatively complex, and the cost of production may be high. And also, if you think about using embryonic cells, it was shown very well that you can activate oncogenes, and if you transplant the embryonic cells into an animal model, up to 80% of these embryonic-derived cells cause tumor formation. Allogenic cells may carry viruses, and especially also synogenic cells may carry viruses that could be potential human pathogenic. Cell culture itself is complicated, and risk of infection may exist because the testing may be difficult on some. So the other alternatives I would like to focus your attention to would be stem cells in your body. So where are the stem cells in your body? We have at least 4 sources of stem cells. And let me mention that they're probably just everywhere. They're called the endogenous stem cells that sit in the skin, in the bone, in the liver, in every type of tissue. But what is our main source? It is the bone marrow, the long tibia, and the long bones that we have here. Cells are recruited in case of injury, and the blood is the bone marrow. The blood is the second communicating system. And then a big source of stem cells comes from your adipose tissue. And the fourth is the first line of defense that sits everywhere in the body. And I will show later, we've identified the cells as being CD90 positive. So apart from a cell culture concept, I would like to focus your attention today to an in vivo concept of tissue regeneration. So making red blood cells, now you may wonder, why would we want to do a regeneration of cells in the body? And you may have wondered how much time it takes to make, for example, red blood cells. A lot of literature exists, and how much time would you take just to count to 7 million? It would take you 7 days just to repeat the numbers, and counting to 20 takes about 6 seconds. But to make 2 million red blood cells is just 1 second. So the human body seems to have an efficient way in your own stem cell bank, which is your bone marrow, to produce your own cells. So why would you want to go to cell culture if it takes such a long time? Now, of course, your body is more than just red blood. It has a number of tissues in the liver, neural tissue, skin, bone, cartilage, and whatever, you name it. Now, in the body we have multipotent stem cells. You can see some examples that I took from the literature. In the bone marrow on the left side, on the heart, on the brain, everywhere we have the cells, but from this multipotent situation, cells must be differentiated going to a commitment to become neurons, muscle tissue, or cardiac tissue, or even blood cells. So the technology, I will introduce you briefly, and we have to repeat a little bit what I showed you yesterday, going into more detail, is a technology that tries to do everything inside the human body, instead of doing it in a cell culture method. And apart from the scientific concept, I will discuss it as a technology to treat acutely injured or degenerated tissue. So I see stem cells like pilots that we have everywhere in the body. We just need to tell them what to do. And the stem cells need a microenvironment. Just like an airplane has a tower that directs the system to allow the pilot to go to a specific direction and to deliver his cargo, the passengers, I believe stem cells can do the job. And our own stem cell factory, the bone marrow, can cope with whatever request we may have. However, we have one problem derived from human nature. We cannot repair big defects. So So the technology that I will show you is in a way the scientific basis how a human being could be enabled by the point-of-care process to repair bigger defects than would be usually expected, and basically comes from preventing scar formation. Now stem cells in big injury, and here's a donor site, which not only would take a long time to heal, a human being has developed a mechanism to cope with big defects by replacing the defected tissue with a scar. But you can see in this experiment, or this study in a human being, left arm and right arm, where an identically sized donor site was taken, this wound has healed in about 6 days, and the other side was treated identically, has not even started. So what we found is a molecule that is released in injury, would be produced systemically or locally, but if the injury persists, it's not available topically, but supplying it topically, you see the wound heals, even though it's a relatively big defect. In this child that had a second-degree burn that was indicated for necrectomy and skin transplantation, we did the same endogenous stem cell activation, treating the child on the third day of the operating. Just 4 days later, a deep second-degree burn that would normally be treated by skin transplantation healed. New skin came up, you can see it, but at 6 months or 1 year later, no pigmentation change is visible anymore. We did a multi-center, double-blinded, placebo-controlled study in Germany with the leading burn centers. Another case that is on a global scale important is chronic wounds. This is a non-responding patient for 3 years that was treated 2 times by endogenous stem cell activation, replacing a signaling molecule that would be made in a healthy skin but is not available in the skin anymore, induces skin closure or granulation tissue formation, as you can see here at 1 week, and that skin was then closed at 2 weeks. The mechanism is based on a stem cell. So if my hand or my fist would be a stem cell, has 2 receptors, one for the local trauma and the second one for a co-signal that's made in the skin. So the gel that we develop is part of endogenous stem cell activation signaling containing synthetic peptides that appear to be responding to a stem cell that's activated in the presence of trauma cytokines. And when the wound heals, the mechanism stops because the trauma cytokine stimulation also stops. This is a venous erosion patient that is treated with this gel that heals in 1 week. The aesthetic application after scar injury, chronic patient, you can see the scar start to remodel and go flatten considerably after 6 days. This is another way, and when we add cells that come from the peripheral blood, we need only 10 cc, then they are prepared and enriched, and they are used for preparing growth factors that come from the patient in a point-of-care bedside procedure in the treatment room with, meanwhile, CE-certified systems that absolutely have no contact to the outside environment. So this is a laboratory equipment that, in a way, is placed into the operating room. And this is an approved medical product. As you just see with the cells, and we put the signals on it that allows such a patient, for example, diabetic wound care patient, to heal instead of receiving amputation. In this case, about 13 centimeters of the heel were missing. In normal human beings, you could regenerate only 2 millimeters of your fingertip, for example, but you can see even this severely infected wound not only is removed, is removing the infection here, but the skin regenerates, but not only the skin, the tissue underneath over a distance of 13 centimeters, including the subcutaneous tissue is still missing here, it comes back. So this patient delivers or receives an anatomically correct shape, almost correct shape of the heel after a long period of time. Unfortunately, it's half a year of treatment, but it shows the safety also. There were no side effects, and we have treated about 50 diabetes patients in this way. Now, the scientific basis is the type of cell. It's a CD90 cell. We published a few years ago. It is a stem cell that is a multi-lineage differentiation capable cell that's prevented in a way from becoming a fibroblast. Other applications in the cardiac field I show briefly. So this mechanical valves that we implanted 28 children in Germany a few years ago already, starting with valves that grow with the child. See here, this is a bioreactor. So the idea is to do a manufacturing in the operating room with a homograph being placed inside, cells are inoculated, and then it is implanted. The system starts to rotate in this patient's preparation area, and you can see it remodels. In vitro takes about 6 weeks, in vivo only 2 weeks, and these were the animal studies that we did a few years ago. There can be also vascular implants, small vascular grafts, and I have to rush up a little bit. I apologize to get it to show to you. These vascular grafts in a control with an approved metal product were always clotted if they were not seeded with the cells. Tissue regenerates in vivo. It takes always an average about 7 to 10 days to create a new graft. We published in Lancet recently a child and airway implant together with Professor Belcher, Martin Elliott, and Professor Valia. And this child has meanwhile grown 12 centimeters. In spinal cord injury, it's the same mechanism. It's prevention of neuronal apoptosis and prevention of cells to go into the fibroblast differentiation, allow a stem cell window to open, and bring in commitment signal not only to create neurons that we show you replicating in vitro, but also neuronal sprouting. So the idea is to transplant it in an in vivo condition. In this case, we had an animal study going on in a paraplegic patient, and 2 days later you see in cases that were the end stage 5 and the severest form of spinal cord injury, the dog is able to stand up in 2 days, but not in a perfect manner yet. It is still having an incomplete response at 2 days and no satisfactory response to pain stimulation. But just about 8 days later, you can see 23 out of 23 dogs walking spontaneously following the severest form of spinal cord injury if the treatment was started within the first 35 hours. So like in heart attacks, stroke, burns, there's a time running after injury. So I believe it's a differentiating mechanisms of progenitor cells going to the fibroblast lineage. And what we do is interruption of this fibroblast lineage, getting another signal, for example, for neurogenesis in vivo that just imitates a microenvironment of a small form of injury. Now in the liver we find the same type of cells, and astoundingly only in the group that has the trauma this type of cells comes up in the body that are known to be fibroblasts, but in reality we don't see any increase in cirrhosis but acceleration of healing. And you see only in the trauma group here, but in the non-trauma group, the control group, the therapy with this peptide structure does not lead to an increase of the cells that just sit around the vascular tree and wait for their duty to be done to participate in bone healing, in liver healing. Or here show you bone healing. Cells alone in this animal study do not do the job. You see the control that we had an in vitro pre-differentiated bone structure that was implanted. It took us 7 days and healed in 16 weeks in the animal study. Now we have reached the stage where we just would take blood, about 20 cc for the patient, inoculate on like calcium phosphate-based material coated with the signaling peptides. The patient, after his implantation, contributes the wound sickling and the bone remodels in 6 weeks. So there's no need for cell culture, which is of course a different approach from a regulatory point of view. I show you some tumor patients that had chondroma removed and the bone regenerated after 1.5 years. Cartilage works the same. We have now in this platform technology about 400 patients in Germany, and the next in line to go to human applications starting from animal studies is the cartilage area. Instead of cell replication in vitro, which is standard practice now, we have a point-of-care process. It takes just about 10-15 minutes to regenerate the cartilage inside this study animals. See, this is a horse filled up with a cell gel if the patient is aporotic, and then it recovers after a few weeks. The distance between the bones becomes bigger. Now, coming to the end of my presentation, for sports medicine, athletics, and people that have tendon injury, for example, in human medicine, there's hardly any form of therapy that we can apply. And this shows a little bit more in detail the process. It starts in a preferred way, just taking blood, which is a non-invasive procedure. In earlier times, we also tried to take bone marrow, but we found we don't need this quantity of stem cells. It's better to activate them locally, because the risk of bone marrow harvesting is not comparable to the risk of just taking a blood sample, which is a small risk and can be extremely standardized. So in this case, the surgeon makes the ultrasound analysis. You can see here the injection into the injured tendon area in ultrasound analysis. You see if the tendon is injured, it's dark and slowly becomes white again. The same we showed in other ligaments. In conventional therapy, it deteriorated, and it is a cross section. You can see here that it comes up in a whitish area, regenerating here after about 8 weeks. Now to the end, what is the scientific concept basically behind it? What is the idea? Basically, Of course, we have a stem cell micro. If you would cut myself here, a wound occurs and signaling starts, and the wound heals with no scar in a few days, if you're lucky. If you're not lucky, we have a big wound, for example, like a burn, and we would get a scar. So you may wonder why this is the case. You as a human being, in seeing yourself in an evolutionary, historic development, think back 100,000 years, if you would have a big wound, we would want to close it as fast as possible to avoid infection. If a heart attack, have a hole, rather than getting a hole into the heart, it would be better to have a fibrotic scar. But genetically speaking, every human being can heal a small defect perfectly. Even a diabetes patient was originally able to do this. So what we try to imitate is a biomimetic process that imitates the microenvironment of a small wound in a big wound condition. And of course, the protocols that we use are totally different because the microenvironment in the brain, the heart, the bone, and the cartilage and liver are totally different. But the basic concept is a biomimetic concept, and the history of bionics starts with Leonardo da Vinci in the European culture to try to design planes like birds. Now we have mechanical implants like hip prosthesis or knee implants, but what I would like to show you, introduce you the ideas of rather to see bionics as a method of communication, like a signaling process. If you remember the picture of the pilot that I showed you before, to get the stem cells into the right niche. Stem cells in this sense probably are important, but by alone they are nothing. They cannot do the job, but the niche control makes the difference in vivo. So it's a technology that regenerates tissues in situ and in vivo. Stem cells are still very important, but stem cells alone cannot do the job. And what we do is just a use of your own genetic mechanisms, of the patient's own genetic mechanisms of repair. So it's a puzzle. I see it like a puzzle where they put different stones together. Many of them have been proven by other colleagues also. There are scientific publications of this myriad and complexity of individual components. But the main technology is a bedside point of care biotechnology and the discovery of the effect that you just need to prevent this type of cells, the CD90 cells, from becoming fibroblasts. That is the scale of intensity. Of course, the wound in diabetics exists or a normal patient, but it may not always heal, but it's a major contributing factor and an old surgical wisdom. So we always use resident local cells, but transplant cells also from bone marrow, blood, or adipose tissue, depending on what kind of application we do. Then we add the boosting molecule, a boosting signal that is like a door opener for stem cells that prevents the transgression of fibroblasts. And then we give another signal, which is the commitment, for example, to make neural tissue, bone, liver, or cartilage. And of course, cells do not grow into empty spaces. We need a scaffold to grow if we would need to repair an airway or heart valve or a piece of bone. So it's a mineral structure or homograft. And last but not least, to make it even more intense, we can use our own stem cell bank in our bone marrow, and we activate the patient's own cells to release chemotactic signals to push up this production inside the human body. Now, how to apply it? What is needed? We have a GMP manufacturing. The idea of pharmaceutical production in the operating room, this is a quality control system, and it's like a standard laminar airflow system that contains a microcomputer that in a way controls the process. So standardized procedure, and why would we want to use spirators? Spirators integrate the materials, position cells, standardize the procedure, and in a way it's an automation technologies of something that was unthinkable of being automated before. So the idea is a wound, different protocols and different materials, bioreactors and processes. So the innovation is twofold. One, the scientific concept, imitating biomimetic processes to induce scar-free healing. On the other side, a translational concept that could make hospitals autonomous by being the producers because it just makes 15 minutes preparation time. So what did we learn about this? And of course, we as human beings also started from 1 or 2 cells. So a whole human being grew out of just a few cells. So I think there's a myth about the number of cells that we would need by cultivating them outside. We just need the triggers aspects that also may have happened during our own ontogenesis. So what is the $6 million man? The bionic idea. I think the changes from bionics to biological bionics instead of technical implants make bio-implants that regenerate inside the body by using the body's own capacity of repair. And this is the acknowledgement to our small animal colleagues, large animal, and our veterinary doctor, Jean-Claude Dianita, And this is my research team from the university that contributed to this scientific work over more than 20 years. Thank you very much.